PU champions? I hope all my dear children are doing absolutely great. So yes, today I am going to solve the model question paper 3 of the year 2025-26. And now you might be wondering, ma'am, first examination is over and now you're giving us MQP? Well, don't worry. Whenever I did revision for the first preparatory examination, I covered all this question there chapter wise. But now as a whole question paper, we will be dealing with it. Okay, so that you can find everything all at once. So I hope you're ready and I hope everyone is super energetic like Jasmine ma'am, right? I am super energetic today because I'm thinking that everyone who watches this also should have the same energy, which is why I thought I should boost up my energy a bit. Yes, so let's go ahead with the first question out there, and it goes like, even in the absence of pollinating agents, seed setting is assured in. That means, suppose there is no pollinating agents, there is no honeybee, there is no insect, there is nothing, there is no wind, water, anything out there. Still, seed setting is there. That means seed is definitely being formed. How is seed found, my dear children? So I hope you guys know that we have an ovary, not us, basically the plant. We also have an ovary, but that's different. Okay. Anyways, the plant has an ovary and then it also has an ovule. Yes. Inside the ovule, you have the embryo sac. Inside that, you have the cute little female gamete. Okay. Female gamete, right? I was going to write male gamete. Now, the pollen grain which carries the male gamete will be coming down over here, fusing with the female gamete and producing what? The zygote. Where is it happening? It is happening within the embryo sac, that is there within the ovule. Now, since zygote is being formed here within the ovule, my dear children, you will not call it as ovule anymore. You will rather call this as a seed. You will call this as a seed. So this is how generally a seed is being formed. Pollination happens, then if at all fertilization also happens. If it happens, my dear children, seed is being formed, and that's how things work. But suppose there is no pollinating agent, still you are very sure that seed is definitely going to be formed. What is the example of this plant? Cucurbita, papaya, commelina, maize. What is the answer? The answer is commelina. You have 3 plants which you need to remember: commelina, oxalis and viola. Commelina, oxalis and viola. I always remember it as cow. So cow with a V. So commelina, oxalis, viola, my dear children, they have 2 types of flowers. One flowers are closed flowers which has the female as well as the male part right over here. So they are closed. The male and the female part are really close, which is why you do not need any pollinating agents. The pollen grains can easily land up like this. And these type of flowers are known as clistogamous flowers. Clistogamous flowers, CL stands for closed. You can remember like that. They are closed flowers in the same plant. So this is more towards the root or the rhizode region. Over here in the picture I depicted it like this, but it is mostly towards the root region or just above the soil. Now there are open flowers as well. Oh, where did it go? Yeah, there are open flowers as well. You call them as chasmogamous flowers. So here also male and the female part will be there. Now they can undergo autogamy, that is known as self-pollination, or they can undergo geitonogamy. They can also undergo xenogamy. That's their choice. But as of now, over here you have been asked, even in the absence of pollinating agents, seed setting is assured in what organism? It is commelina, because over here definitely autogamy is going to happen. Self-pollination is going to happen anyhow. Done? Done, ma'am? Clear. Next question. Which of the following statement is true? Okay. LH surge induces menstruation. LH surge. So when LH, the amount of luteinizing hormone increases, you might have all studied that basically there is follicle. Inside the follicle you have the secondary oocyte. When the LH hormone increases, this follicle will be burst opening. Okay, this follicle will burst open, and what is going to be released? Basically, basically the secondary oocyte, which is right over here, it is going to be released. What do you call this? This is known as rupture of Graafian follicle. So rupture of Graafian follicle happens and the secondary oocyte is released, and this process is known as ovulation, not menstruation. So this statement is wrong because here it says that LH surge induces menstruation. Wrong, ma'am. Now, regression of corpus luteum increases progesterone. You know what? You have corpus luteum. Okay, so basically we were here, right? Ovulation happened. Then what happens to this Graafian follicle? Whatever is left behind, what happens to it? Basically, it is still there with another name. You call this as corpus luteum. What do you call this? Corpus luteum. Ma'am, this corpus luteum is there, but why is it still there? What does it produce? So it produces 2 very important female hormones. One is progesterone. One is progesterone and the other one is estrogen. But majorly, little partiality is given to progesterone. And when the level of progesterone is increased, you know what happened? It is a hormone that maintains our endometrium. Not just maintains, it makes sure that the endometrium is fed very nicely and it is very thick now. It is very thick now. Okay, so that is the role of progesterone. Estrogen also does it, but progesterone does it mainly. But you know what? Progesterone, when it increases in amount, usually this thing happens in our life also. Whenever we achieve everything, we think that we are the boss of everything. But that's not how it exactly works. So we need to respect the roots from where we came from, right? Similarly, this guy didn't respect. Okay, so progesterone said that I do not like LH, I do not like FSH. Please stop their secretion. I cannot handle this. So LH and FSH, my dear children, when they are present, especially LH, when it is present, that's only when corpus luteum can be maintained. Only in the presence of LH, your corpus luteum can be maintained. So as of now, it went a surge and then it came down, but still there, and it's still maintaining what? The corpus luteum out there, and the corpus luteum is releasing progesterone. But now the progesterone says, I cannot see LH secretion over here, please stop it. Now if it is stopped, obviously corpus luteum is not going to be maintained. If corpus luteum is not there, progesterone also will not be there. You get it? And the progesterone is now sad. So regression of corpus luteum increases progesterone? No, baba. Regression means if this particular thing is disintegrating, is going away. Do you think progesterone will be there? No, 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 no. Wrong, wrong, wrong. Next. Diploid egg is formed after second meiotic division. Diploid egg. The egg is what? The female gamete. Can any gamete be diploid after meiotic division? So after meiosis, my dear children, everything becomes half in number. Okay, it becomes half in number. And that too after second meiotic division? Yes, definitely. So this is a wrong statement. It is a wrong statement. It has to be haploid egg. Half number of chromosomes will be there. Now, embryo differentiates from the inner cell mass of the blastocyst. So, you know, embryo formation, when it happens, it's not a one-step process. There are multiple stages of embryo. And one such stage is known as blastocyst. And it's a very cute stage. I'll tell you why it's cute. So, over here, suppose this is the blastocyst stage. You have an outer layer, you call this as trophoblast. What do you call this? Trophoblast. And then inside that, you have a mass of cells, which you call as inner cell mass. Now what do you expect? You expect that all of it will become an embryo, later a fetus and things like that. But that does not happen. Even though this is an embryonic stage itself, you call this as an embryo itself. But what exactly happened is that this part of the blastocyst, that is the trophoblast, will be completely utilized for the formation of a layer that is known as placenta. So from the baby's side, you have to contribute something, and from the mother's side also some contribution happens, and that's how placenta is being formed. So from the embryo side, you have the trophoblast being there for the formation of placenta. Now who will become actual fetus? You need an actual fetus to be formed. One has went to make the placenta. So what is left behind? You have the inner cell mass. So we say the inner cell mass which is left behind is something that is going to differentiate into actual embryo later on. Okay, into actual embryo later on. So this statement is correct. You had to find what is true. So embryo differentiate from the inner cell mass of the blastocyst. This, ma'am, is very, very true. Okay? Okay, yes. So statement number 3, statement number 1, question number 3, statement number 1. GIFT is an in vitro fertilization technique. So first of all, what is GIFT? GIFT stands for gamete intra-fallopian transfer. Inside the fallopian tube, you are transferring gamete. Inside the fallopian tube, you are transferring gamete. Why do you think a gamete is being transferred inside the fallopian tube? Probably because the person whose body this is, my dear children, she might not be able to produce good amount of ovum, or the ovum might have some defects, or ovum are not being produced at all. Gametes are not being produced at all. But she has a perfect uterus such that if at all copulation happens and the sperm comes and fuses, she can give rise to a whole baby out there because it's a very suitable environment. It's just that she is not able to produce gametes. So why not gift her gametes? And that is known as gift. Gamete intrafallopian transfer is where you take the gametes from a donor who is able to produce it. You take the gametes from the donor and you put it inside the fallopian tube of this particular female who is not able to produce gametes. Now what is going to happen? Not able to produce gametes, or is not producing the good quality gametes out there. Anyway, I'm putting out there. Why up there? Because fertilization has to happen in the fallopian tube itself. It's a natural process. The sperm has to come over here in the fallopian tube and undergo fertilization. It will form a zygote, then form embryo. The embryo will roll down, come over here, get implanted into the endometrium layer when the embryo in its blastocyst stage. We just discussed, right? So that's how it usually works. So here, do you think in vitro fertilization happens? In vitro means inside the laboratory fertilization is happening. Fertilization means sperm and egg being fused. It is happening inside the laboratory? No, Baba, it is happening within the female body. So this particular statement that you see over here will be wrong. Right. Now statement 2 says in gift gametes are fertilized outside the female body. We just discussed it right now. It cannot be outside the female body. It is within the female body. So both the statements, my dear children, are wrong. Okay, both the statements. So while we do the questions, we expect at least one statement to be correct, right? So over here, both of them are wrong. Both statement 1 and 2 are incorrect. Don't go tick correct. Okay, fine. Now the next statement that we have is gene located very close to one another on the same chromosome tend to be transmitted and are transmitted together. Okay, transmitted together and are called as. What do you mean by that? Basically, suppose I have chromosome over here. Okay, this is A chromosome, this is B chromosome. Basically, they are very close to each other. Suppose I want to sap these regions of the chromosomes. What will happen? Now I have new chromosomal combination where A and B have successfully come over here. You know, they went together. Why did they go together? Because, my dear children, they are tightly linked genes. They are very close to each other. You call them tightly linked genes. And suppose they are far away from each other, you will call them loosely linked genes. In that case, if at all they are loosely linked, things would be quite different. So suppose A is here, B is here, and here you are swapping. After the swapping, what will happen? B will be still here, lower part is being swapped, okay, A will go there. So this means they are loosely linked. They are loosely linked. They have less, closeness among themselves. So over here it is written, gene located very close to one another on the same chromosome tend to be transmitted together and are called what? Linked genes, Baba. No allelomorphs, no identical genes, no recessive genes. It is what? Linked genes. Understanding? My dear children, are you getting it? So whenever we say like this, the same gene is going out there, we say the amount of recombination, new combinations being formed is less. So tightly linked genes will have less recombination, new combination, whereas loosely linked genes like this will have high amount of recombination. Okay, done, deal? Yes, ma'am. By the way, are you liking the video? If at all you're liking the video, then what are you waiting for? Share it with your friends and click on the like button. And yes, my dear children, if at all you are someone who is preparing for KCET, what have we launched? We have launched your most favorite ABHYAS test series. You had been asking us for a very long time from the starting of the session actually. I think it's because of the effects of these people who went and told the other children as well that this particular series is really, really good. So the previous time we launched it in a thought that this is definitely going to help you out, and you know what, the proud moment came up when almost thousands of children got very good ranks, and among them we could meet a few of them. Okay, there was time and everything before they went to vacations, so we all met. So Prajwal with rank 171 out of 3 lakh children who gave the examination. That's not a small deal. Kiran with rank 443 could not meet him, but yes, Diksha with 693 met her. Sachin with 934 didn't meet him, but Mohammed Sahil, we met him. He was having 2427. He was a person who was writing NEET, this, that, so many things. And my dear children, you know what, when you have the determination, you will be able to do it, and with the right resource. And this is one such right resource. And we have 35 KCET mock tests, but apart from that, we have added up one more thing for your benefit, and that is chapter wise KCET PYQs. So once your chapter is done, that is this, you can go ahead and solve some questions of KCET also and see what all do you know, what are the areas where you lack. And you know what, we didn't change the price is the same. It is just rupees 1499. So giving you extra benefit because we know it is definitely going to help you. And my dear children, over here you have all the subjects coverage, that is PCMB, that comes in your KCET examination, and there will be real-time exam pattern as well as timing. So you need not worry about anything out there. We have got you covered. The link will be there in the description section. Go take it up. Okay, now question number 5. Match the following: Alec Jeffrey, F. Sanger, Jacob Monod, and Griffith. Who who, discovered what? That is the question. So you have these many options: lac operon, transferring principle or transforming principle actually, annotated DNA sequencing, and DNA fingerprinting. I'll tell you easy way of remembering this thing. So Alec Jeffrey, FF is there, okay? FF is there. Basically, he was the person who went with DNA fingerprinting. F. Sanger has a S over here, and this S is for DNA sequencing, annotated DNA sequencing. Jacob and Monad, basically both of them has O in it, basically, so it is lac operon, also starting with O. Operon all starts with O. And Griffith, my dear children, we all know he did the transforming principle experiment with the little mice up there and Streptococcus pneumoniae. This is how I generally tend to remember, but yes, if at all you have the time and space to remember it as such, please go ahead and remember it like that. Okay, so Alec Jeffrey, DNA fingerprinting. F. Sanger has an S in the second word. Similarly, all of them, if at all you see the second term, you will be able to find that as well. So Alec Jeffrey, DNA fingerprinting. F. Sanger, annotated DNA sequencing. Jacob and Monad, lac operon, and Griffith, transforming principle. It's not transferring, it's actually transforming principle. I do not know if it's a typo by our team, by my team, or basically it's there in the MQP like that only, but anyway. So your answer for this question becomes what? It becomes this: A with 4, B with 3, C with 1, and D with 2. Okay? Yes, ma'am. Next question. Observe the following diagrammatic representation of operation of natural selection on different traits. Here, natural selection depicts what? So what does natural selection depict over here? You have a graph where 2 peaks are there. Well, when you have 2 peaks, it shows up that 2 extreme characters are being chosen. It shows that 2 extreme characters are being chosen. For example, I say that in this crowd, there are people who are tall. And there are people who are dwarf as well. So this means that I have 2 graphs. Suppose I am choosing only the tall one. So there will be only in one particular side. Only in one particular side. I'll say that this is a directional one. Either of the one are being chosen. Either of the ones are not tall. That side basically means it is dwarf, right? So this is dwarf. This is directional natural selection. This is what? Disruptive natural selection. Now you have one more where neither tall nor dwarf is taken up. It's just the medium character which is taken up. That is medium height. So that will be what? Stabilizing graph. Stabilizing graph. So among them, if at all you see, you have 2 peaks. 2 peaks means disruption is happening, not stabilization, not directional. It is what? Disruptive. Then cannot be depicted. Yes, it cannot be depicted if at all you guys not study. But you are studying right over here. You're good children. So which is why the answer for this question becomes what? C. Now you would have this thought, my dear children, that second preparatory examination might come a little bit later. I'll do after some time. I know this thought is very natural to come, but please don't, let this thought conquer your mind, and do not be delayed, do not do procrastination. Please start studying. That's all. Okay? Yes. Now, which of the following plants does not possess hallucinogenic properties? Does not possess hallucinogenic properties. You know what is hallucination? You start seeing things which is actually not there. For example, I start seeing ghost. Okay, not exactly, but yes. Or I see cows flying. I always give this example. I don't know why. Cows flying, and then a mouse very big like me. So basically, it's like that. That is known as hallucination. Things which are not there, you tend to see them. Now, there are few plants which has hallucinogenic properties. For example, Erythroxylum coca is one such plant from where you get cocaine. From where you get cocaine. Now, this Erythroxylum coca is native to South America, and my dear children, it gives you cocaine. Please remember, coke is also known as coke or crack. But anyway, in high doses, it can cause hallucination. Atropa balladina is also a hallucinogenic plant, and Datura is also a hallucinogenic plant. Datura picture is also given in your NCERT. It looks something like this. It looks something like this. This is Datura, my dear children. That is also one more very important thing. Now, Papaver somniferum is papagai because papa, okay, papa in Kannada means pavan, no? So basically over here, Papaver somniferum is responsible for production of opioids, which has nothing to do with hallucination. They are usually like heroin or morphine. Heroin or morphine. Morphine is a sedative and a painkiller. That means you feel to sleep after that, and also it's a painkiller. It is given after surgeries by doctors to the patients who have underwent surgeries. Heroin is a depressant and it slows down your body function. As I said, it has nothing to do with hallucination, so this one is the odd guy out. So the answer for this question becomes D. Okay? Yes, ma'am. Next question, question number 8. Choose the correct statements. Innate immunity is accomplished by providing different types of barriers. Innate immunity is something that is there with me from my birth. That means, for example, have you seen anyone born without skin? Have you seen anyone who is born without tears? No, right? It is something, very rare cases, okay, about tears. But every one of us has skin, my dear children. Skin comes with our birth, and this is a type of immunity. You call it as innate immunity, something that is there from us, with us, from our birth. Now, there are different types of innate immunity. As it's been said over here, there are different types of barriers under innate immunity. The skin that I spoke about comes under physical immunity. Okay, physical barrier, not physical immunity, is physical barrier. Then you have physiological barrier, where you're talking about mucus secretion, tears, everything. They are all physiological barriers because they also have some enzymes. And these enzymes can potentially kill any bacteria and anything out there. Okay, so that's why it is a physiological barrier. Now you have cellular barrier. Some cells are there with us from our starting, and they are there for fighting with other foreign particles out there. Then you have cytokine barriers, where certain cells will be releasing something that can protect other cells. Example is interferon. Okay, you might have seen, your question usually comes, a virus infected cell is there. What is this particular cell releasing? It releases interferon. Interferon is something that's saying that, hey, other cells, please be safe, please be safe. There is a virus attack happening up over here. So those type of things actually comes from birth, and my dear children, it is very non-specific. It is non-specific. What do you mean by non-specific? As in, your skin does not know that it has to, basically, if a virus comes, if a bacteria comes, if anything else comes, our skin is going to be neutral up there. Does it have any specification that, okay, I will let you in, you are not allowed? Does it say like that? No, obviously. So there is no specific type of immunity out there in innate immunity. Now there is something opposite to innate immunity that is known as acquired immunity. Something that develops within us through infections, through vaccinations and things like that. We basically acquire it during our lifetime. Inherit is innate immunity. Acquired is something that we acquire throughout our lifetime. Here, different types of things become active. For example, your B cells, your T cells, B lymphocytes, T lymphocytes, all of them become active. So I know it's too much for you to remember, so we'll go one by one. So innate immunity is accomplished by providing different types of barriers. Yes, physical, physiological, cellular, cytokine barriers. Okay. Now acquired immunity is present from birth and is inherited from parents. Arey Baba, it's not. It is not at all inherited from parents. It is not present from birth. We acquire it throughout our lifetime. Now acquired immunity can be divided into antibody mediated and cell mediated immunity. Can it be divided into antibody mediated and cell mediated? Yes, my dear children. So basically what we have is that in acquired immunity there are two type of things which are super active. What are those two type of things? One is your B lymphocytes, another one is your T lymphocytes. Both of them are immunological cells. B lymphocytes, when it comes and sees some antigens or some pathogens within our body, they are going to release some specific antibodies against it. Some specific antibodies against it, and apart from that they will also release some memory cells. These memory cells have an idea that, okay, this time this thing happened, the next time I have to release the same antibodies but in more amount. That is the use of this. Okay, and now these antibodies which are there, it has been mixed with, listen, these cells are there in our blood, and now these antibodies are also going to be mixed with our blood. So we say it provides humoral immunity. It provides humoral immunity since it is mixing with our humor. Humor is nothing but a fluid in our body. So blood is a fluid. That is why. Now T cells which are there, they are very active. They do not produce antibodies. They go directly kill some other cell which is having some problem, or they directly kill the pathogens out there. That is what you mean by T cells, or at least they help somehow with the help of the cell production that they do, which is why I call this as cell mediated immunity. Cell mediated immunity. So acquired immunity can be divided into antibody mediated and cell mediated immunity. Exactly. Why? Humoral immunity is just antibody mediated immunity only. Now innate immunity is also called specific immunity. I told you it's not at all specific, so that is wrong. Acquired is specific, innate is not. Now acquired immunity, this is fifth point also, okay. Acquired immunity consists of specialized cells, that is T cell and B cell, and antibodies that circulate in the blood. Does it? Acquired immunity, does it contain all of this in the blood? Yes, my dear children. Yes, obviously yes. So this fifth statement is also correct. Somehow I missed putting it down. So fifth statement is correct, third statement is correct, first statement is correct. With that, we get to understand that C is my correct answer. C is what? My correct answer. So I mark them over here so that you don't have a confusion, as well as over here. Now, question number 9. During sewage treatment, the gas produced by anaerobic sludge digester includes. Now, if at all you are someone who has no idea about what is anaerobic sludge digester, just understand, whenever you're talking about sewage treatment and whenever you're talking about gas being produced, it has to be biogas. It has to be biogas. And biogas is always produced by anaerobes. That means the organisms that does not need oxygen to survive. They will not survive under oxygen. Basically, they do not need oxygen to survive. What are they producing? Biogas. And your biogas is made up of three very important things. One is your methane. The second one is hydrogen sulfide. The third one is carbon dioxide. 1, 2, 3. 1, 2, 3. So methane, hydrogen sulfide, carbon dioxide, I have my answer, that is C. If at all in your question it is not hydrogen sulfide but just hydrogen, do not worry, that is also correct. But they won't give you an option, they won't give you both the options together. Either of the one you will be getting it. But please understand, if instead of hydrogen sulfide they are mentioning as hydrogen, it's not wrong. H2, it is not wrong. Okay, biogas made by anaerobic bacteria, which is a common bacteria that we have, methanobacterium. We have methanobacterium. Cool? Yes, ma'am. Now, question number 10. Which of the following is not the feature of plasmid? Which of the following is not the feature of plasmid? What is plasmid? So, suppose I am talking about one E. coli. Say hi to E. coli. So, E. coli, my dear children, will be having its own genetic material for sure. Okay, will be having its own genome, which is super important for the production of proteins, which will be letting it survive. But apart from that, this guy will also have some extra chromosomal thing. Extra chromosomal as in, that is not very important for the production of protein that will help it survive and all. No, it's just there. It is extra chromosomal out there, and usually it is found in double strand. Not usually, it is found in double-stranded nature. So extra chromosomal, they are double-stranded, and whenever E. coli replicates, the genome also replicates. This guy also replicates. Why? Because it has its own origin of replication. Only if at all you have origin of replication, then only you can replicate. Just like how we have Aadhaar card, then only we can live peacefully in India. Similarly, it also needs origin of replication. Got it? Yes. So extrachromals, correct, ma'am, which is not the feature is being asked. Okay. Circular structure, correct, ma'am. Single stranded, no, it is double. Single, ready to mingle. Okay, so it is double stranded, and independent replication is obviously being seen. Why? Because origin of replication is also there. Got it? Yes. Clear? Crystal clear? Done. Now next, Agrobacterium tumefaciens is used as a vector for cloning genes in. Agrobacterium tumefaciens is a very big name. Why? Okay, is used as a vector for cloning genes in. Vector means it is someone who carries something very important. That is vector. That is a gene of interest for that matter. Okay, it carries my favorite gene of interest for that particular matter. Now, Agrobacterium tumefaciens, whom do you think it's acting as a vector for? That is a very important question. Plants, animals, bacteria, viruses. Plants, animals, bacteria, viruses. So I'll tell you something about Agrobacterium tumefaciens. So this is the bad bacteria that we have. Okay, this is a very bad bacteria what we have. Why? Because basically this guy, my dear children, has a tumor-inducing plasmid. TI plasmid stands for tumor-inducing plasmid. It causes tumor in the case of plants. How exactly? I'll tell you. So basically over here you see in this plasmid there is a region which you call as tDNA. What do you call this as? tDNA, or you also call this as transfer DNA. So basically when this bacterium goes and affects the plant, it passes on this tDNA. Basically the plasmid eventually gets entry into the plant cell, and while plasmid is going, obviously the tDNA is going. The tDNA causes problems in the particular plant. It causes tumor in the plant. So that is how it usually works. But now we human beings are basically more clever. Okay, clever. So which is why we did one very important thing. We thought we'll remove this tDNA. We remove this tDNA, the transfer DNA, and anyway, it has a basic tendency to go and infect the plant. No, why not infect the plant? But this time with our gene of interest, we put our gene of interest over here, and now the same organism will go and affect the plant, but this time what is it passing? It is passing the gene of interest. So your answer for this question would be what? Plants. Not animals, not bacteria, not viruses, it is just plants. In the case of animals, who is the usual organism that affects the animals the most and uses their machinery to replicate in everything? It is retrovirus. HIV, you know, HIV is a retrovirus. So basically here, if at all you're talking about animals and you need a vector, it has to be retrovirus. Make sure that this retrovirus is what? Disarmed. It is disarmed. That means any pathogenic property that it has, it is being removed. It is safe to be used. Okay, so over here that is not the question. The question is about agrobacterial tumefacients, and I hope you will remember about the plants. Now going ahead, identify the incorrect statement. Transgenic cow Rosie produced milk enriched with alpha-lactalbumin. What is transgenic cow? We are making some genetic changes in the cow. That's why you call this as transgenic cow. Probably we inserted a gene that we wanted into the plant, into the organism out there. Now this cow, whose name is Rosie, very cute, very cute name, right? So basically produced a milk which is enriched with alpha-lactalbumin. Do you think it's true? What is alpha-lactalbumin? It is actually a human milk protein. It is very nutritious, it is very important, and now we are making a cow produce it. Do you think that's possible? Yes, we did. So this is a correct statement. This is not an incorrect statement. Next, Bt toxin gene scribe 1AC controls corn borers. We'll come to that. C statement. Human protein alpha-1 antitrypsin is used to treat emphysema. So there is another human protein just like alpha-lactalbumin. We have another human protein alpha-1 antitrypsin. This particular protein is used to treat what? Emphysema. Emphysema is what? A respiratory disorder. It's a respiratory disorder. Now if at all I make an organism produce this alpha-1 antitrypsin, basically I can use it for the treatment of emphysema, right? Do you think it was possible? Yes. With the help of the transgenic animals, that was also possible, and this is correct. You need this for treatment of emphysema. Now D option. The commercial production of human insulin was made possible by the transgenic, basically, by the help of transgenic E. coli. That is the question. Okay, the commercial production of human insulin. Now humans produce insulin. What is the use of insulin? Insulin basically makes sure that our blood glucose level is less. How? So it converts all the glucose to something else which is known as glycogen. And the insulin is produced by what? Insulin is basically produced by pancreas. Okay, pancreas of our body. Now suppose insulin is not being produced enough in someone's body. Obviously in their blood the glucose level will be so high, the sugar is high, we say they have diabetes. Right. Now my dear children, can I make an organism produce human insulin? Initially, insulin of pig, cow and everything was given, but people tended to have what? Allergies, which is why it was thought that we can produce human insulin with the help of E. coli, and that was done by a company known as Eli Lilly. So this statement is also correct. Now what we are left with? We are left with this statement B, which is obviously has to be incorrect. So Bt toxin gene scribe 1AC controls corn borers. Is this a correct statement? So my dear children, always understand, first of all, what do you mean by Bt toxin? So just like agrobacterial tumefacients, you have another bacteria that is known as Bacillus thuringiensis. Okay, Bacillus thuringiensis. This particular bacteria that we have contains protoxin or prototoxin, basically inactive version of some toxin. Who is responsible for production of this particular toxin? It is nothing but the gene which is known as cry gene. There is a cry gene that produces prototoxin. Now it's an inactive version, it does not kill the bacteria obviously. Now suppose a caterpillar had this bacteria by some chance. Now inside the midgut of this particular caterpillar, what is going to be happening? The Bt toxin, the prototoxin which was there, it is going to convert into active form of toxin. Now you might be thinking, ma'am, bacteria does not have brain, how does this happen? So basically in the midgut region, the bacteria gets all the necessary environment for the activation of this prototoxin. There is an alkaline pH up over here. there is what? An alkaline pH in the mid-gut of the bacteria, which is why this prototoxin converts into what? Active version of toxin, my dear children. So over here, if at all your question is beta toxin genes cry 1AC controls corn borers, how will you say? Yes ma'am, yes ma'am. No. So now you have to know that the cry genes which are there, they are very specific. What are they? They are very specific. These are cannot just randomly act on any insect out there. They are very specific. So over here, remember, you have 3 cry genes in your NCERT. One is cry AC, 1AC, and one is cry 2AB. First letter starts with C. Second letter starts with B. This is how I remember. So cry 1AC, cry 2AB is for killing cotton bollworm. They release toxin that kills cotton bollworm. Now you have one more thing, that is cry 1AC is done, cry 2AB is done. Cry 1AB is not done, right? So we need one. So cry 1AB is over here. You just need one term, where the first letter is C over here, corn, and last letter is B, that is borers. So both of them starts with C and B, but this is a bigger term, which is why you need 2 things. Over here, you just need one thing. You just need one thing, and starting should be C, ending should be B. That's how I remember it. That is for corn borers. Now over here, cry 1AC is being written. Cry 1AC is actually for cotton bollworms. So can I say this particular statement is wrong? Obviously, ma'am, you can say that. So yes, this particular statement is wrong. Got it? Yes. Now, the correct order of various levels of biological organization in an ecosystem. This is one of the easiest question out there. So what is the correct order of various levels of biological organization? Organisms comes together to form population. Population forms to community. Community forms ecosystem. Ecosystem forms what? Biome. And hence, I have got my answer. A is my answer for this particular question. Rest everything has ulta things. Okay? Next. The rate of the formation of new organic matter by the producer is the rate of formation of new organic matter by the producer. Listen, we have the producer, we have the primary consumer, we have the secondary consumer. Suppose this is grass, has to be rabbit, this has to be tiger. Now over here I am saying rate of formation of new organic matter. First of all, since it's a producer, primary producer, I will call this as primary production. Whenever it produces something, I'll say it's primary production. And when I have to find the rate of it, I'll use the term productivity. So it will be what? Primary productivity. Your answer for this question will be primary productivity. Now there might be a question, ma'am, why is it not gross primary productivity? Because in our NCERT it is written the rate of production of organic matter by the plants is known as gross primary productivity. That is when you talk about multiple plants in an ecosystem. In an ecosystem, the total of organic matter being produced by the first trophic level is known as the gross primary productivity. Now all of it is not given to the next organism, right? It is being produced, but some of it is utilized for its own purpose, some of it is lost. Okay, so I'll say respiratory losses. Now whatever is left behind is known as NPP, that is net primary productivity. Again, you talk about an ecosystem out there. Over here, you're talking about an individual producer, which is why you just say primary productivity and you do not use the term net primary productivity or gross primary productivity. The same thing if at all it is done by the rabbit. What is the new rate of formation of new organic matter by the second organism in the second trophic level? It will be known as secondary productivity. Okay, got it? So please understand it. Next, as per species inventory, 70% of species recorded are. As per the species inventories, 70% of the species recorded are what? Plants, fungi, bryophytes or animals. 70% of the species, the highest organisms out there. It will be animals. It will be animals. Under animals, if at all we go ahead, 70% of the animals are insects. Or what? Insects, my dear children. Always remember that. Fungus is not. So fungus, basically I can say the fungi is actually too many. Okay, fungus is basically a combination of fishes, amphibians, reptiles and mammals put together. That is the amount of fungus. So three things are given in the NCERT. 70% of all the organisms are animals. In that 70% is insects. And that if at all you're talking about fungus, fungus is a total sum of fishes, amphibians, reptiles and mammals. That is FAM. Okay, yes. And now we go to the favorite part of the question paper, and that is what? The fill in the blanks. Zygote divides to form dash, which is implanted in the uterus. Implanted means it goes settle down in the uterus, endometrium of the uterus. Now you tell me the answer for this. Otherwise, obviously I wouldn't do that. So zygote divides to form what? What stage of embryo? Blastocyst. First of all, go through this option. Neanderthal man. Neanderthal was the basically first organism to cover their body and bury their dead organisms out there, or the dead relatives out there, right? That was Neanderthal. Decreases primary lymphoid organs, Clostridium butylicum, and increases is given. Okay, fine. Now we can easily find. 17th question. Stage in human evolution used hides to protect their body and bury their dead. It is what? Neanderthal man. 17th. 18th question. Dash are the site where immature lymphocytes differentiate into antigen-sensitive lymphocytes. That means immature lymphocytes, as in you're talking about B and T lymphocytes, they're immature, they don't know what to do, but now they know, they become antigen-sensitive. That means they get to understand that this is my dushman, I have to kill it. But it's not start killing it, it just becomes sensitive to it. When you say it has started to kill it, you call it as effector cells. Does not become effector cells in the primary lymphoid organs. Primary lymphoid organs are the answer for this question. They are the site where the immature lymphocytes differentiate into antigen-sensitive lymphocytes. That's all. In the secondary lymphoid organs, they are going to do dishum. They are going to become effector cells. So your answer for this question would become this. Now, dash is a producer of butyric acid. Butyric acid is Clostridium butylicum. Easiest question. It is what? Clostridium butylicum. So that is a 19th question. Now, in general species diversity, dash as we move away from the equator towards the pole. Now, from the equator towards the pole, if you move, pole is something that will be having frequent glaciation, lot of things, and now the glaciers might melt also. There is no time for the species to adapt to all this changing environment, which is why the species diversity is going to go less towards the poles. Equator, it will be having more, but towards the pole, it will be less. So can I say it decreases? It decreases as we move from the equator to poles. But in this type of questions, please make sure you pay attention to details. Equator towards the poles is given over here. Poles towards the equator, and then you write decreases if you come, right, like that, and you come. Basically, what is going to happen? You're going to lose your marks, nothing else. Okay? Yes. Next, now we are going ahead with the 2 marker section. You have to attempt 5 of the questions, but I have explained all the 7 questions. Listen to me carefully. We'll go fast over here. Okay, so let's go. Micropyle contains a small, micropyle remains as a small pore, not pole, it is small pore in the seed coat of the seed. So if at all you see, my dear children, this is the ovule. Ovule has 2 integuments. The open area which is over here, you call this as micropyle. So when the ovule converts into a seed, still the micropyle remains that as a small pore. Why do you think it remains like that? So that exchange of gases can easily happen. So that what? Exchange of gases, water and oxygen during seed germination can really happen. That is the reason. So micropyle of the ovule persists even after fertilization and become a tiny pore in the seed coat. Why? Because it helps in the entry of water and oxygen during seed germination. When the seed has to germinate, it needs water and oxygen. That is being done with the help of this. Now there is one more part to the question, that is apple and cashew are not called true fruits. Why? Why that partiality with them? So usually what happens is that when the ovule becomes a seed, the ovary becomes a fruit. Agreed? Now in some plants, the other parts of the plant also help in the formation of fruit. It's not just ovary, but the other parts of the plant also helps. For example, in the case of apple, you know, you might have seen the plants usually rest on a thalamus like this. This thalamus also contributes for the formation of fruit in the case of apple. Now in the case of cashew, you know, this pedicel which is there, the stalk which is there, that also helps in the formation of fruit. So these type of fruits which are not just formed of ovary, but the other inflorescence of the flower, you call this as false fruit. Got it? Yes, ma'am. So, true fruits develop only from the ovary after fertilization. In apple and the cashew, the edible part develops from the floral parts other than the ovary. Hence, they are called false fruits or accessory fruits, not true fruits. They're not called as what? True fruits. Got it? Yes. The next question. Mention any two events that are inhibited by the intake of oral contraceptive pills to prevent pregnancy in human. Mention any two events that are inhibited by the intake of oral contraceptive pills. So whenever someone has oral contraceptive pills, what are the things that are being inhibited? First of all, you should know oral pill contains two hormones, progesterone and estrogen, or it can be progesterone or estrogen, either one or both of them together. Now they are super important in our body for the process of ovulation to happen. You know, ovulation, LH surge is being caused. Who causes it? It is estrogen that causes it. Now if at all ovulation is not happening, that is a problem. Now implantation. For implantation you need endometrium. Who technically maintains it even more better? It is progesterone. So basically, usually they do all this thing. But when we are causing a hormonal imbalance in our body by giving them from outside resources, okay, aren't we giving them in the form of oral pills? Our body is already producing it. You are giving it. It creates an imbalance, which is why the normal functions that they used to do, ovulation they used to do, implantation they used to do, they're not going to do it smoothly right now. And which is why we can prevent conception to happen. Okay, we can prevent the baby formation to happen. Okay, so ovulation, they stop the release of ovum from ovary, and implantation is something that they again prevent. They make the uterine lining unsuitable for the implantation of the fertilized ovule. I hope you got it. Fertilized egg. Okay, not ovule. Now identify the symbols used in the pedigree analysis. A square is given, a circle is given, and two lines are given. Whenever two lines are given, that means it is consanguineous meeting. That is meeting between relatives. Two lines, that means more bond is there. You can remember like that. Usually we just have one line like that shows meeting. But if you have two lines, that shows consanguineous meeting or meeting between relatives. Now you have a square, a circle, and then again you have square. This square shows that, this square which is there, it shows that there's a male child, but it is being colored also, which is why it shows that it is an affected male child. That means this particular male child is there he will be having what? A particular disease. That is what you mean by a square with a colored thing. Got it? Yes. Now next thing. Draw the labeled diagram of nucleosome. How do you draw the labeled diagram of nucleosome? First of all, what is nucleosome? Nucleosome is a structure onto which your DNA wraps around. Why do you think we need to wrap around our DNA? So basically, you know, we are eukaryotes. We have nucleus inside our cell, and our DNA is very big, 6.6 into 10 to the power 9 base pairs long. So we need to fit it inside the small nucleus. Okay, so we need to make it compact, and for that we need the help of certain very important proteins. These proteins are known as histone proteins. And how many of them are there? There are almost 8 histone proteins. 1, 2, 3, 4, 5, 6, 7, 8. 8 histone proteins are there, and on this histone proteins wraps around what? Your DNA. Okay, DNA wraps around it. How many times? It wraps around twice. It wraps around twice. Okay, now 8 histone proteins are there, DNA is wrapping around twice, and my dear children, you have one more histone protein right up over here. You will call this as H1 histone. H1 histone is a separate thing, but it is there. This whole thing which is there, it forms a very important structure which you call as nucleosome. What do you call this? You call this as nucleosome. And this is a diagram that you have to make. Remember the DNA, how many base pairs of DNA are being covered over here? Almost 200 base pairs of DNA are being covered right up over here. Done? So this is one very important question. Now we have the next question out there, and that is question number 25. Question number 25, state the evolutionary relationship giving reasons between, someone came in, okay. State the evolutionary relationship giving reasons between sweet potato and potato. Or basically state the evolutionary relationship between sweet potato, and please give reasons to it. That is the question. So basically, potato is how many people's favorite? First of all, it's my favorite. I love potato. I can eat as many potato as I want. But no one gives me potato. Nor do I make potato. So basically, potato which is there and my sweet potato which is there, my dear children, both of them are quite different. So I'll tell you the story, okay? So we have 2 plants. And this plant, the stem of it goes down underground and basically forms some storage units like this. And do not be worried, it has its own root also. It has its own root as well. Now there is another plant, obviously 2 plants, and over here you have the roots. The roots are basically modified to form something. So here there is a stem modification, here there is a root modification. This is your sweet potato. This is what? The sweet potato. Okay. And over here, this thing that you see, my dear children, is a stem modification where the stem is used for storage, and this is known as just the potato. This is potato. So sweet potato and potato is there. So state the evolutionary relationship giving reasons between sweet potato and potato. Sweet potato is a modified root, whereas potato is a modified stem. Both store food but differ in origin as well as structure. Okay, both of them are going to differ in origin as well as the structure. Such organs with same function but different origin are called analogous organs. What do you call them? Analogous organs. So basically, they have the same function of what? Storage. The function is same. Function is same, but if at all you see structural similarity, they are not structure. One is stem, one is root. So structural similarity is not there. Okay, structural similarity is not at all there. You call this as analogous organs. And why do you think analogous organs are there? What is type of evolution that is happening up over here? Both of them are having to store their food over here underground, my dear children, because probably their habitat is forcing them to. Their habitat is probably forcing them to. So can I say that both of them converge to a point and are doing the same function? This is showing convergent evolution? Yes. So A for analogous organs, and basically they are showing convergent evolution. As simple as that. And clear? Yes. Going ahead. Discuss the role of fungi as a biofertilizer. Fertilizer means what? Basically, they increase the nutritional quality of the soil. Okay, they add some nutrients out there. How can fungi act as a biofertilizer? I'll tell you. So basically, there is a fungi which belongs to the genus. It belongs to the genus Glomus. And this fungi, my dear children, they can go seep into the soil. They can actually go seep into the soil and absorb very important things that even plant roots cannot go directly absorb. That is phosphorus. They can, but the amount will be very, very less. But the fungi can do it very efficiently. Okay, not just that. They don't just provide phosphorus, and hence it is known as biofertilizer. But apart from that, let me tell you, they also helps in increasing the resistance and tolerance. Resistance to what? Resistant to any pathogen that can be formed in the roots. Or tolerance to what? Tolerance to salinity. As in, if there is too much salt content, still the plant will be able to survive because of this. Tolerance to what? Tolerance to drought as well. Even when water is not there, it can easily survive. So increase of resistance increases, and also tolerance also increases. Apart from that, overall increase in the plant growth and development is also being managed by this particular fungi. So biofertilizer is there. Apart from that, a lot of other functions are also there. Now you might think, why is the fungi doing all this? Because, my dear children, the root is basically attached to a whole plant. A whole plant can do photosynthesis and can provide some organic matter to the fungi as well. So it's a symbiotic relationship. This is also an example of mutualism, where both of them are mutually helping down each other. Okay, so role of fungi as a biofertilizer, I hope you have got it. So it does not help in increasing the yield or anything like that. Overall growth and development, increase in resistance and tolerance, that is all is there. And also absorption of phosphorus. Apart from that, nothing else. Now, write any 2 advanced techniques used in ex situ conservation. And I have written the difference between in situ and ex situ conservation, which is why this particular question is wrong. So let me correct it. And let me give you the answer. What are the advanced techniques used in ex situ conservation? So usually what do we have in ex situ? Ex situ means conserving them outside their natural habitat, right? Yeah, I should not be cutting that, right? Conserving them outside their natural habitat. Now usually we have zoological parks, that is zoos, botanical gardens for the plants and everything. If you talk about advanced techniques, we have a few of them. Listen to me carefully. One of them is cryopreservation. cryopreservation. What are you doing over here? You are basically storing the gametes in minus 196 degrees Celsius in liquid nitrogen. So basically making sure that the gametes of the plants which may be extinct are also safe, or the animals which may be extinct are also safe. That's cryopreservation. First thing. Second thing, the plants, in them the seed banks are there for storing the seeds of the plant which are soon going to be extinct, or they are in endangered species. Now, apart from that, what do you have? You have tissue culture. Basically, again, plants which are soon going to die, I make a culture out of it. I do micro-propagation. Multiple plants are being formed with the help of tissue culture. This is another advanced technique. And now in the case of animals, we have IVF, in vitro fertilization. Nowadays, you might have seen couples tend to have babies later on. So what do? They freeze their eggs so that in vitro fertilization can be done. Okay, within the lab fertilization can be done, or later on this particular egg can be put inside the female body, either way. But anyway, in vitro fertilization is also a technique where male gametes and female gametes are taken and they are made to fuse within the laboratory condition. This is also making sure that the species, my dear children, if at all the egg and the sperm is being safe, the species can be going on continued. Okay, so these are the advanced techniques of CO2 conservation. There are 4 of them: cryopreservation, seed bank, tissue culture, and also IVF. Done? Done. Now, question number 28. This is from part C. Draw a neatly labeled diagram of a typical anatropous ovule. And just give me a second. I'll be right back. We are right over here. So, draw a neatly labeled diagram of a typical anatropous ovule. How do you make it? So, first of all, we have to make the ovary. Just have an understanding of how a normal ovule looks, and then you'll have a better understanding on things. So you have an ovule, and the ovule, my dear children, are covered by 2 integuments. Okay, 2 sweaters up there, and it's been opened, unbuttoned over here. This region is known as micropyle. Cool. Opposite end of micropyle will always be chalazial end. Fine. Now over here, you would be having placenta. And this placenta is there for nourishment of the ovule. And how is it attached to the ovule, my dear children? With the help of a very important structure, which you call as funicle. So you have the placenta and you have the funicle as well. And the point of attachment right up over here is what you call as hilum. Cool. This is the general structure. Now suppose you are changing the position of placenta from here to here. Okay, there's a shift in the position. There is opposite sides up there. What exactly happens? It will look almost like this. The placenta, the funicle, as well as the hilum. The chalazial end is always the opposite end of the micropyle, so it remains the same, and that is the picture being shown over here. So this broad thing that you see is the funicle being attached at the point which is known as hilum. You have 2 integuments out there, and this opposite side is known as chalazial end. The first side that we have, the open side, is known as micropylar end. That's all. Okay, this is the structure of the typical anatropous ovule. Now question number 29. Name the pituitary and the testicular hormones involved in spermatogenesis. Name the pituitary and testicular hormones involved in spermatogenesis. So FSH, if at all you say, that is one hormone, and the second hormone that we have from the pituitary gland is LH, luteinizing hormone. Apart from that, if you talk about testicular hormones, which testis releases, my dear children, you have testosterone. And how do they function? How they are involved in spermatogenesis? Listen to me carefully. So first of all, let's make the pituitary gland. You have the pituitary gland. It releases 2 hormones. One is LH and the other one is FSH, follicle stimulating hormone and luteinizing hormone. Now you need to understand one very important thing. If you talk about the testis, inside that you have testicular lobules, and inside that you have seminiferous tubules, right? Suppose this is a seminiferous tubule, this is also a seminiferous tubule. Here you have certain cells which you call as Sertoli cells. And here you also have certain other cells which you call as spermatogonia, something that will form sperms later on, but only if nourishment is provided by Sertoli cells. Now in between 2 seminiferous tubules, you have a space where over here you have certain cells which you call as Leydig cells. What do you call Leydig cells? They are the one that releases testosterone. And now we're going to see how do they actually release testosterone. So LH, luteinizing hormone, which is released by the pituitary gland, is going to act on the Leydig cell. and the Leydig cell, my dear children, instead will be releasing testosterone. It will be releasing testosterone, that is the male sex hormone, right? Now FSH, which is there on the other hand, is going to act on the Sertoli cells. And Sertoli cells are there for the nourishment of the sperms or the spermatogonia out there, right? The Sertoli cells instead is going to release some very important factors. Now you might think, why do you need these factors? That is one very important thing. So listen to me carefully. When the whole process of spermatogenesis happens, that means from spermatogonia being formed, it differentiates into primary spermatocyte. This undergoes meiosis. You get what? Secondary spermatocyte. This undergoes the second round of meiosis. You get what? You get spermatids. Spermatids are round globular structure. They are round. They're not like sperms. You need to convert these spermatids to spermatozoa. Now this whole process is known as what? It is known as spermatogenesis. Whole process is known as spermatogenesis. In that specifically, the conversion of spermatids or the transformation of spermatids to spermatozoa is known as spermiogenesis. Okay. Now you need testosterone throughout this particular process, which is why what exactly happens is that testosterone, which is produced over here, slowly enters the seminiferous tubules. So it enters the seminiferous tubules. Now you have to make sure that it's there throughout, such that the whole process of spermatogenesis smoothly happens. And that is exactly where you need the factors, because these factors are basically androgen binding factors. Testosterone is an androgen, and this is androgen binding factors. Make sure the androgens have been bonded up and they are staying upright over here, such that the whole process can take place smoothly, because without testosterone this process cannot happen, especially the process of spermiogenesis, especially the process of formation of spermatozoa that needs high amount of testosterone right up there. So these two are pituitary hormones, and this thing which is there, my dear children, it is the testicular hormone up there. So FSH, follicle stimulating hormone, secreted by the anterior pituitary gland. So pituitary gland has two lobes. One is anterior, one is posterior. Anterior lobe produces FSH and LH. FSH acts on the Sertoli cells on the seminiferous tubules and stimulates spermatogenesis. Basically, production of some very important factors which help in spermatogenesis. LH, also known as ICH, interstitial cell stimulating hormone. It is secreted by, again, the anterior pituitary gland, acts on the Leydig cells of the testis and stimulates the secretion of testosterone. Now testosterone, which is testicular hormones secreted by the Leydig cells of testis, essential for the initiation and the maintenance of spermatogenesis as a whole, especially for the formation of spermatozoa. This is known as spermiogenesis. Cool? Yes, ma'am. Now question number 30. What is medical termination of pregnancy? Mention any two medical grounds on which pregnancy can be terminated. What is MTP? Medical termination of pregnancy means you are doing abortion. And this is not a natural case of abortion. This is intentional abortion or induced abortion, that is known as MTP, medical termination of pregnancy. Now this can be done using giving medicines or basically during surgical, but either way, it has to be done only under two medical grounds, and that is being asked. What are the two medical grounds? First, when it is risked the life of the mother or grave injury to her physical or mental health. Second one, when there is severe congenital abnormalities or genetic defects in the fetus, something that has been inherited from the parents to the offspring, or something that comes up as a genetic disorder in the baby. If it is risk to the mother's life, it is risk to the baby's life, then in that medical ground, you can take what you call MTP to be done. Now, MTP was first legalized in the year 1971, and because people were using it for their own benefit, as in they started doing amniocentesis, they started knowing the sex of the child. If it was a female, they started doing MTP. So female feticide was increasing way too much, which is why an act was being put forward, and that is Medical Termination Amendment Act in the year 2017. So legalized in the year 1971, but the act was put in the year 2017. And my dear children, it was said that if at all someone is doing MTP in 12 weeks, that is basically 3 months duration, you need the approval of at least one medical professional. If at all it is being done in 6 months time period or 24 weeks, you need at least 2 medical professionals, agreement out there. and it is only safe up to 12 weeks. It is only safe up to 12 weeks. Apart from that, it is not considered to be safe, even though it can be done up to 24 weeks, but it is not considered to be safe. You have that in your head as well. Okay, so these are the few things that you need to remember about MTP. Going ahead, Australian marsupials and Australian placental mammals explain convergent evolution and adaptive radiation. Justify this statement. So Australian marsupials and Australian placental mammals, how do they show convergent evolution? You need to know that. How will you get to understand that? If at all, I explain you well. So listen to me carefully. Suppose there's an Australian marsupial that came up over here in one particular island. Okay, in one particular island. Some of them moved towards this side. He moved here, some of them moved here, some of them moved here, some of them moved here. All of them got adapted there. Okay, this is an example of adaptive radiation. So they radiated and basically they got adapted at each places out there. This is an example of divergent evolution, where you have a common ancestor and basically you're diverging to different locations and you're getting adapted there. This is an example of divergent evolution when you talk individually. Now suppose you're talking about placental mammals. They also came over here. Placental mammals came over here. Some moved this side, some moved this side, some moved this side, some moved this side, some moved this side. Again, adaptive radiation. Individually talking, they also show divergent evolution. But now if you talk about both of them together, you do not have any common ancestor. But basically, since both of them are living in a common area, they will have some similar functions. Even though the structure is different, they have similar function. Analogous organs. Analogous organs, structure is different, internal structure is different, but similar function because they are habitating in the same area. So can I say it is convergent evolution? They are converging at one particular point over here. That is why it is an example of convergent evolution. So when both of them are considered, I say convergent evolution. If individually, then it's divergent evolution. So they will be having obviously some common factors out there. So Australian marsupials, in Australia, many marsupials evolved from a common ancestor stock and diversified into different forms, adapted to different habitats and functions in a geographical area. They moved around. This is an example of adaptive radiation. Agreed. Placental mammals in the Australia evolved independently, but developed structures and habits similar to that of the marsupials which were there and occupied the similar ecological niches. This similarity is due to what? Convergent evolution. Now the example is given right over here. They have made a list and this list is super important. In the other model question paper, we solved the question also. So mole is there from placental mammal side, Australian marsupial side, you have marsupial mole. Anteater is there, you have numbart, which is another type of anteater from that side. For all you say the structural similarity, they are quite same. Mouse, marsupial mouse is there. Lemur, spotted cuscus is there. Flying squirrel, flying phalanger is there. Bobcat, Tasmanian tiger cat is there. Wolf, Tasmanian wolf is there. Always remember here on this side you will have smaller names. Okay, placental mammals, you have smaller names and most of them are alternative names, rhyming mole, marsupial mole and things like that. Anteater and numbart is different. Lemur and spotted cuscus is different. Flying squirrel and flying phalanger is different. Rest all of it has cat, wolf, and all of it. Okay, so please go through it. It can also be a question. Now, question number 32. Name the causative organism of the following disease. Ascariasis. Ascariasis, my dear children, is caused by a worm that is known as Ascaris lumbricoides. Ascaris lumbricoides, right? Ascaris lumbricoides. And ringworm is, my dear children, caused by fungi, trichophyton, epidermophyton and also microsporum. Microsporum, trichophyton and also epidermophyton. Now typhoid you have. Typhoid is caused by Salmonella typhi. Salmonella typhi causes what? Typhoid. Cool? So these many things are right up there. Okay, easy question. Now mention the use of following bioactive agents. Streptokinase, what is the use of it? Streptokinase, it's an enzyme. AS is there, so it's an enzyme. Where do you get it from? Streptococcus. And what does it do? You know what? Suppose someone is having myocardial infarction or heart attack. Why is it happening? Because probably their heart is not receiving enough amount of oxygenated blood out there because there might be some clots in there. Clot might be there. There should be someone to burst down the clot. And who is that? It is streptokinase. It's an enzyme that is known as a clot buster as well. It busts down the clots. Now you have cyclosporin A. You get cyclosporin A from a fungi which is known as Trichoderma polysporum. Suppose my kidneys are not working fine. I am taking kidneys from you. You are ready to give me kidneys, okay? And now I have it in my body. What usually happens is that my body senses that this is something foreign. I have to reject it. This thing can cause problem in my body. Is what my immunological cells think, because it is very foreign for them. So they go react to it and they cause problem. So that it leads to rejection. I don't want that to happen. So I want to suppress my immune system, which is why I would have what? Cyclosporin A, which is an immunosuppressant. It is a immunosuppressant. You get it from a fungi, that is Trichoderma polysporum. Now you have statin. Statin, you get it from another fungus which is Monascus purpureus. And what does it do? Statin, I say it stabilizes. Statin stabilizes. What does it stabilize? It stabilizes the blood cholesterol level. It stabilizes the blood cholesterol level, my dear children. Okay, so statin is also super important. You get from another fungi, that is Monascus purpureus. Okay, yes. Stabilizes or basically lowers down the blood cholesterol level. That will be a better way, but I remember as stabilizes because statin, stable, it is more easy to remember. Next, ecological pyramids have limitations to justify with 3 reasons. So ecological pyramids which are made, my dear children, producer is there, and then you have a consumer, then you have a top consumer and things like that. Anywhere over here do you take decomposers? Decomposers are super important. When an organism dies, everything important is leached back into the soil, my dear children. Who does that? Decomposers. But do we take their name over here? No. So they do not consider decomposers, which is very wrong. This is one such limitation. Second thing, is it always like, suppose I have a plant over here and plant is fed by rabbit. Rabbit is eaten by tiger. Suppose there is deer. Will the tiger eat only the rabbit? Deer can also be eaten. So there can be multiple food chains that work. Basically, can I say there are multiple food webs which are there? Yes. So they assume a simple food chain, whereas most ecosystems have what? Complex food webs. Okay, there are complex food webs out there. Cool? Yes. Now, they cannot represent organisms that occupy more than one trophic level. For example, human beings. Human beings can eat plants. Human being can eat animals also. So in that sense, I can be either a primary consumer or a secondary consumer. I can be either a primary consumer or a secondary consumer, right? So multiple trophic levels are being occupied by a single organism. That can also happen. But here, can we show that? No. Okay, so they cannot represent organisms that can occupy more than one trophic level. So that is also one very big problem. For example, omnivores. And that's how we end up part C, that is the 3-marker question. Now we have the 5-marker questions up there, and the list goes like this. Double fertilization is a unique event taking place in the flowering plants. Briefly explain. How do we explain double fertilization? So listen, this is your embryo sac. Inside the embryo sac, towards the chalazal end, okay? Chalazal end is an end where nothing important happens. You can remember like that. So we have 3 cells out there. You call them as antipodal cells. Okay, antipodal cells. Now over here in the micropylar end, listen, it is micropylar end. You have 3 cells. 2 synergids, and one very important cell which you call as the egg cell. All of it together, I call this as the egg apparatus. Synergids and the egg cell, I call this egg apparatus. They are best friends. Now you have one big cell right over here which has 2 polar nuclei. What does it has? It has 2 polar nuclei. So I'll change it and I put it in a different color. 2 polar nuclei. So this is having N. All of them are haploid, but this big cell having 2 polar nuclei, that will be diploid in nature already. Okay, N, 2N, right? Now double fertilization means 2-time fertilization has to happen. So you know what? One pollen grain which comes and attaches, it has 2 male gametes. One male gamete which is there, it will fuse with the egg cell. One male gamete fuses with the egg cell, and what do you get? You get the zygote. Male gamete also has haploid thing, egg cell is also haploid, haploid comes together, you get a diploid zygote. This process is known as syngamy. This is known as syngamy. First fertilization is done. Again you have a male gamete. This male gamete comes and fuses. The male gamete comes and fuses with the 2 polar nuclei over here of the central cell. This cell which is there, which is having 2 polar nuclei, we call this as a central cell. The male gamete is having N, this guy also has N, this also has N, N, 3 times 3 nucleus fuses, which is why I call this as triple fusion. But what do you get? You get PEN, penna, primary endosperm nucleus. This is known as primary endosperm nucleus, and the cell which is going to be formed, we'll call this as primary endosperm cell. 2 times fertilization is happening, one is syngamy and the other one is triple fusion. The primary endosperm cell later on divides and forms the endosperm as a whole. So double fertilization is a unique event taking place in flowering plants. Briefly explained, this is the explanation that I have to give you. Cool? Now, arrange the following terms in correct development sequence. Pollen grains, porogenous tissue, microspore tetrad, and the pollen mother cell and the male gamete. How do you think, so we hear about the female and the male part, both of it is being asked. So how do you say? You have the microspore mother cell, or you also call this as the pollen mother cell. The pollen mother cell undergoes meiosis 1 and you have division. Okay, it gives rise to haploid cells out there. Again they undergo division. You have 4 cells at the end. N. Basically, they are microspores, and you know what? They're all close together. I call them as microspore tetrad. I call them as microspore tetrad. You do not say microspores, you say microspore tetrad. Then this microspore tetrad dissociate from each other, and that's how you get the microspores. That's how you get the microspores. These microspores later form the pollen grains, and the pollen grains will be having the male gamete. So over here, your answer for double fertilization goes like this, and arrangement goes like this. Porogenous tissue, okay, first you have sporogenous tissue. That's a group of PMC cells or MMC cells. That forms a tissue, right? Now pollen mother cell, then microspore tetrad, then pollen grain. The pollen grain has male gametes. Okay? Okay, ma'am. Next. Draw a label diagrammatic view of the female reproductive system. Over here you have to draw like that. Oviduct, make sure you mention isthmus, ampulla and infundibulum. Infundibulum has finger-like projection known as fimbriae. Over here you have the ovary. This is the uterine fundus. You have 3 layers in the ovarian wall, uterine wall. One is, outer layer is perimetrium, then you have myometrium, and the innermost glandular layer that we have is endometrium. Right over here we have cervix, this canal which is there, cervical canal, and then you have the vagina. Got it? Yes, this is a diagram that we have to make. It's an easy diagram, right? Comparatively easy. Now represent schematically the result of one gene interaction between pure tall pea plant and the pure dwarf pea plant. You have to talk about one gene interaction. That means you're talking about the monohybrid cross. You're talking about the monohybrid cross. So how do you show that? So over here, capital T, small t, small t. Okay, so this is pure tall and this is pure dwarf. Now over here, these are the parents. Now you need to form the gametes. Gametes will be capital T, here small t, small t. Now you make the F1 generation, the first generation. This and this crosses, you get capital T small t. This and this crosses, you get capital T small t. This and this again capital T small t. This and this capital T small t. All of them, my dear children, will be tall. Why? Because capital T is going to be dominant over the small t. Right? Now I need to confirm that if at all it is, so we know the plants are tall, but he didn't know, the Mendel didn't know whether it is tall like this or it is tall like this. For that again he did the cross, he did selfing. So in this case the parents will be what? The parents is going to be capital T small t and again capital T small t. What is the gametes going to be like? Capital T small t, capital T small t. Now you fuse them. This is the F2 generation, filial 2 generation. So when this and this crosses, you get capital T, capital T. When this and this fuses, you get capital T, small t. This and this again capital T, small t. This and this small t, small t. So if at all we see, one capital T. Okay, this is tall. Capital T, small t, again this is tall because capital T is there. Capital T, small t, again this is tall, and one is going to be dwarf. So 3 of them are going to be tall and 1 of them, my dear children, is going to be dwarf. This is what the phenotypic ratio, the physical appearance ratio. 3 tall, 1 dwarf. What is genotypic ratio? What is the genetic makeup? So 1 capital T, 2 capital T, small t, 1 small t, small t. So it will be like this. So I'll write capital T, small t, and small t, small t. This is going to be what? My phenotypic ratio. The genotypic ratio, that is genotypic ratio, and this is going to be my phenotypic ratio. Always remember this. Got it? Clear? Crystal clear? I hope yes. Now, enumerate the characteristics of a genetic code. So what are the characteristics of a genetic code? If at all you see all the genetic code, my dear children, they are in the form of three codons. So the codon is triplet. The codon is how? Triplet in nature. In total we have 64 codons. How many codons are there? The list is like 64 codons are there. Out of that, 61 of them codes for amino acid. But, I'm sorry, just a second. Okay, 61 of them, I'll just check this also. Okay, 61 of them codes for amino acid, but 3 of them are not going to code for anything. They are known as stop codons. They are known as stop codons. Now, if at all you see AUG has 2 functions. AUG is going to code for amino acid that is known as methionine, and it is also a start codon. It helps in initiating the process of translation to happen. So AUG has dual function. Now, some amino acids are coded by more than 1 codon. For example, if at all you see leucine. Leucine is coded by CUU, CUC, CUA, CUG. This feature of an amino acid being coded by multiple codons, it is known as degenerate feature. Degenerate feature. But you also need to understand 1 codon codes for only 1 amino acid. CUU is not repeated anywhere else. 1 codon, 1 amino acid. This shows unambiguous nature of the codons. This shows unambiguous nature of the codons. Please remember that as well. Now the next thing, if at all you see an mRNA sequence, there is no full stop, nothing like that. So it is read in a contiguous fashion. It is read in a continuous fashion. And my dear children, you have 3 stop codons. I already told you those are the exceptions. It is UA, it is UA, UGA, and UAG. UA, UA, UGA, and UAG. AG means fire. You can remember like that. UAG is there. UA, and then UGA. Listen, don't show this video to anyone else. No, you should show this video to everyone else because everyone needs to study. Yes. So anyways, the code is nearly universal. What do you mean by that? Basically, CUU codes for leucine over here. CUU codes for leucine in the case of a bacteria also. So we say it is universal. The codon codes for the same amino acid everywhere. Exceptions are there always. Exceptions are what? In the case of some protozoans. In case of some protozoans, and also, my dear children, you know, in the mitochondria also we have DNA. In mitochondria also transcription, everything is happening. So in the mitochondrial codons, my dear children, there will be some exceptions. Mitochondrial codons and also protozoans, they are exceptions. Okay? Yes. Now, name the group of genes that have been identified in normal cells that could lead to cancer. They found in normal cells, but they can lead to cancer. These genes are known as proto-oncogenes. They are known as proto-oncogenes, my dear children. So proto-oncogenes, they normally function, they produce proteins and everything like that. But suppose there is some mutation happening up over here. There is some mutation happening over here. What happens? They are going to basically lead to cancer. They will turn the cells into a cancerous cell. So name the group of genes that have been identified in a normal cell that could lead to cancer. It is proto-oncogenes. How do you write the answer? Like this. Proto-oncogenes. Simple. Now, which technique are useful in detecting cancers of internal organs? You have biopsy and histopathology. Basically, biopsy and histopathology means, suppose over there you cut a small part of the tumor and then you do your studies under whatever tools that you have. That is known as biopsy. Biopsy basically means cutting off, and histopathology means studying it off. Okay, that is the meaning. Now just give me a quick second. I'm getting a call. So that is what you mean by biopsy as well as histopathology. And now you have CT scan. CT scan basically means computerized tomography, where again you are using X-rays to find the three-dimensional images of the internal organs to detect tumors. So over here, please understand, you are using X-rays. Apart from that, you can also have radiography, where basically you're directly using X-rays. That is a different thing. There you do not get a three-dimensional image. Over here in CT scan you get, and that is specifically for internal organs to detect the tumors. Now we have MRI, which is considered to be obviously comparatively safer. Why? Because over here you are using strong magnetic fields and also non-ionizing radiation. What you are using? Strong magnetic fields and also non-ionizing radiation, my dear children. That is the use. So here no X-rays are there, and it is specifically for finding physiological changes in the living tissue. Physiological changes in the living tissue. So that is one very, very important thing about it. Okay. Now these are the few things, and also radiography is also one thing. Now one more question is there, why are cancer patients often given alpha interferons as a part of treatment? So alpha interferons, my dear children, they are biological response modifiers. They make your immune system really, really strong. What is it? Biological response modifiers. So cancer patients, we know a lot of their cells are being dead because of chemotherapy, radiotherapy, a lot of things like that. So you need immunological cells. You need immunity out there. That is why we have the alpha interferon, which acts like a biological response modifier. Done? Yes. Now next question. Explain the application of biotechnology in the field of medicine. Explain the application of biotechnology in the field of medicine. So here comes the role of insulin that we were talking about. So listen to me carefully, my dear children. So just give me a quick second. Yes. So we are here, and listen to me carefully. So suppose I have insulin. You know, insulin which is there, it is made up of A chain as well as B chain, and over here you have a different chain which is known as C chain. This is found in the pro-hormone version, as in the immature version of insulin. Insulin is a hormone, and this is found in the immature version of the hormone. Now suppose I have genes coding for all these things. In our body, what exactly happens is that when this is being formed, the A and the B chains are made to have some disulfide bonds, and the C chain is being removed. The C chain is directly being removed with the help of our machinery. Now suppose I want an organism, that is E. coli, to produce this. I cannot expect the E. coli's machinery to cut off the C peptide. So then Eli Lilly came into the picture. Eli Lilly, which is a company, it came into the picture and said, why to do all this drama? Why not take the gene for A, take the gene for B, and produce A and B chains directly, and why not join them in the laboratory conditions? So this is exactly what they did. They had E. coli. They put A gene in one, B gene in another. A chain was produced, B chain was produced, and both of them, my dear children, was joined using disulfide bonds in the laboratory condition, and that's how you got the first human-like insulin, which is known as humulin. So this is pro-hormone, okay, not a protein, hormone. Pro-hormone, and that is the mature version of the hormone. Cool. So that is one very important thing. Biotechnology helps produce safe, pure and large quantities of human proteins using microbes. Example, human insulin, that is humulin. Earlier, insulin was extracted from cattle, pig pancreas, caused what? Allergies. Later, human insulin consists of A and B polypeptide chains linked by disulfide bonds. Eli Lilly created separate DNA sequences for A and B chains and inserted them into E. coli plasmids. Bacteria produced both chains, which were purified and chemically combined to form a functional form of insulin. Got it? And now the next use that we have is gene therapy. Gene therapy, for example, suppose again, there was a 4-year-old child, my dear children, and this 4-year-old child did not have this particular enzyme in her body. This stands for adenosine deaminase. It stands for adenosine deaminase. This is a super important enzyme. Now this enzyme makes sure that in the cell there is no toxic buildup. And this enzyme is super important. Okay, this particular enzyme which is there, it went. This enzyme is super important in the case of ADA, is super important in the case of what? Lymphocytes. That is some cells which are very active in our immunity. Now suppose I want a cell to have this ADA gene, or I want a cell to produce the ADA enzyme out there. What can be done? I can refer to gene therapy. Gene therapy, where exactly what is being done. Suppose this is the patient whose name is PU2. Okay, from PU2, first of all, what do is that we take lymphocytes. First thing, these steps are super important. You might be asked about the correct order of steps also. First of all, we extract the lymphocytes from the body. The second thing, we make sure that the lymphocytes are cultured. We increase the number with the help of what? Culture method. Now after that, we make sure that with the help of a retrovirus, okay, with the help of some retrovirus which carries our gene of interest, that is the ADA gene, we make the thing infect all the cells out there, and which is how the cells have now got the ADA gene. Now what do? We insert it back, we put it back to the patient. And this is known as ADA gene therapy, where basically we are providing the gene which is responsible for the production of this particular enzyme out there. Now the catch of the story is that obviously these lymphocytes which are being inserted will die after some time. So you need to do repeated transfusion. That is the catch that you have. If at all someone wants to treat ADA, you have to treat it in the embryological stage itself. In the embryological stage, if at all you treat it, it's all good. Otherwise, not good. Got it? Yes. So, gene therapy corrects genetic defects by inserting functional genes into the patient cells. Example, ADA deficiency, first success in the year 1990. ADA deficiency is caused by deletion of the adenosine deaminase gene, leading to an impaired immune system. It will be an impaired immune system. Now, treatment steps. Lymphocytes withdrawn from the patient. Functional ADA, cDNA, ADA as in adenosine deaminase, cDNA, complementary DNA gene or complementary DNA sequence is inserted using a retroviral vector. Genetically corrected lymphocytes return to the patient. Now before that, you also do culturing that comes between the first and the third step. So culturing is also important. You culture the lymphocytes, increase the number. Now this improves immunity, but repeated treatments are needed as lymphocytes are not immortal. They will die after some time. Permanent cure is possible by only introducing ADA gene in the embryological stage or in the stem cells. That's all. Got it? Yes, ma'am. Now, name the type of interaction seen in each of the following example. Clownfish living among the tentacles of the sea anemone. So, finding Nemo fish, right? It is going to stay in the tentacles of the sea anemone. Tentacles of the sea anemone provides protection to the clownfish. Clownfish gives anything back to the sea anemone? No. So basically, can I say that this is getting the benefit, this is neither getting benefited nor getting harmed? So what type of relationship is this? This is known as commensalism. This is known as commensalism. Now, koel and cuckoo bird during the breeding season. So cuckoo basically, so koel and cuckoo, it's the same thing. Basically, koel and crow, my dear children. So over here, the crow lays its egg and the cuckoo also comes and lays the egg. Now the crow will come, provide nourishment, provide warmth and everything to all the birds. So basically, crow's energy is being lost. It is being overutilized by this koel. This is known as brood parasitism. Over here, one is getting the benefit, other is getting harmed because its energy is being lost, raising not its own child but someone else's. You get it? So this is known as brood parasitism. Always remember, it is the koel who have some evolution happening in them, which makes sure that it resembles like the crow's egg. It is not the host but the parasite which undergoes evolution. Always remember that. Now, barnacles growing on the back of the whale. On the whale, you might have seen some barnacles sticking onto them. Over here, the barnacles, basically, when they are stuck on the whale, the barnacles are easily able to move around to different areas. Whale does not get any benefit, nor do they get harmed. Again, this is an example of commensalism. Marine fish and copepods. So marine fish is there. On the surface of it, you have copepods, and these copepods are actually feeding on the marine fish. So it's a parasitic relationship where one is getting benefit, another one is getting harmed. This is what? Parasitism. Now we have sexual deceit in Mediterranean orchid, that is Ophrys. So Ophrys, which is there, it's an orchid, and my dear, it looks like the female honeybee. It looks like the female honeybee. So the male honeybee comes and tries to copulate with it. It's known as pseudo copulation. It's known as false copulation because actual copulation is not happening. This is not a female bee. But anyway, in this process, pollination is going to happen. When it copulates, my dear children, all the pollen grains are going to get stuck on the surface of the male honeybee. Now, wherever it goes next, it will pass on that particular pollen grains. So, over here, it is basically mutualism. Why? Because anyway, while coming over here, it pseudocopulates and also takes down certain pollen grains for itself, for its nourishment, the honeybee. Okay, and on the other hand, the plant is obviously undergoing what? It is undergoing pollination as well. So this is an example of mutualism. Well, this thing is a very bad thing. It's known as sexual deceit, sexual cheating being done. Okay? Yes. Now categorize the following as either autosomal or sex-linked disorders. Hemophilia, my dear children. Hemophilia is a sex-linked recessive disorder. Under sex-linked, you do not have any dominant disorders. You just have recessive disorders. One is hemophilia. Other one is color blindness. Rest all of them are autosomal recessive disorders. Rest all of them has to be autosomal recessive disorders except myotonic dystrophy. Myotonic dystrophy is the only example of autosomal dominant disorder in our NCERT, and it is not written. It's basically the pedigree chart which is made, and below it is being written. So autosomal, everything is recessive. Okay, two of them are sex-linked recessive. One is hemophilia and color blindness. Rest all of them are autosomal recessive, and one example is myotonic dystrophy of autosomal dominant disorder. And for that, pedigree chart is being made. Clear? Now, what conclusions will you arrive after careful observation of the below given diagram? This diagram is given over here. Now, at the sixth position, you see GUG is there, and GUG codes for valine. What does it show? Basically, substitution. It shows substitution. So, initially it was GAG, but the A was replaced to U. And basically, this codes for glutamic acid. It codes for glutamic acid, but over here, this is coding for what? Valine. It is coding for valine. Okay, so substitution reaction happened, which is why one particular amino acid change, and if one particular amino acid change is of the polypeptide chain, the whole protein structure will obviously get disoriented. So this is the chain of beta hemoglobin, which is going to get disrupted. Beta globin chain is going to be disrupted, and if beta globin chain is disrupted, obviously it's a part of hemoglobin. Hemoglobin will also be not of good quality. And which is why the hemoglobin is there in the RBC will also be not in the good quality. It will turn sickle shape in shape. Sickle in shape. Okay. Now identify the disorder by the following symptoms. Individual with overall masculine development but with gynecomastia. So overall, if at all you see, my dear children, there is a masculine development, but they have gynecomastia, as in the breast formation. So overall masculine development with breast formation, it has to be Klinefelter's syndrome. Klinefelter's syndrome. What exactly happens over here? So instead of having just XY, you have an extra X. This is a trisomy condition. You have an extra X. This is known as Klinefelter's syndrome. Got it? Yes, ma'am. Done. So this is the answer that I had given you. Yes, you can go through each of it as well. You will get the PDF. Okay? Join the WhatsApp channel. That's super important. Cool. Now next. State the arrangement of different genes of lactose operon in E. coli. Describe the role of lactose in the regulation of lac operon. So listen to me carefully. This question is super important. It's super easy also. You have to just state the arrangement of different genes. So first thing, this is how the genes are being arranged. There is promoter region. There is an inhibitory gene. What gene? Inhibitory gene. What does it code for? It codes for a repressor protein. Okay, not inhibitory gene, actually a regulatory gene, but over here it's functioning as an inhibitor. So inhibitory gene, and it's coding for what? A repressor protein. Okay, repressor protein. Now here you have another gene, promoter region for that, operator region for that, and these are the three genes that we have. ZYA, ZYA, you can remember like that, ZYA. Z basically codes for beta caroteridase enzyme, Y codes for permease enzyme, and A codes for transacetylase enzyme. Okay. Beta-galactosidase is an enzyme that is going to break down lactose to glucose and galactose. Suppose E. coli is something that has it, and E. coli does not have enough amount of energy. So it needs an energy source to break down, to bring out energy, and that is lactose. So lactose can be broken down to glucose and galactose, and that glucose can be used for energy source. So beta-galactosidase does that. Permease makes sure the permeability of the membrane is done. Okay, it makes more permeable, makes the membrane more permeable. Trans-acetylase, which is there, deals with anything toxic that comes up during the entire process. Fine. Now, suppose lactose is not here. Suppose there is no lactose in the medium where we are growing the E. coli. So do we need these enzymes? No. We do not need these enzymes. Obviously, beta-galactosidase is not required because there is no lactose to be broken down. There is no need for why? Because permease increase the permeability such that the lactose can get inside, if at all it is outside. So we do not need that. We do not need trans-acetylase as well. So basically, we do not need any of these enzymes. I do not need transcription of any of these enzymes. So what do? Basically, the repressor protein which is there, it will go and bind on the operator region. It will go and bind on the operator region. What is the use of it? Now, RNA polymerase which comes and binds on the promoter, will it be able to move ahead? Will it be able to move ahead and undergo the process of transcription? No, it would not be able to do that. And if it is not able to do that, do you think transcription happens? No. So I say in here, lac operon is in switched off condition. What condition? Switched off condition. Now suppose there is lactose. Suppose there is lactose, and this lactose, my dear children, enters the cell. This lactose quickly goes and binds on the repressor protein. Now the repressor protein is not free to go bind up there, because why? The lactose is bonded to it. So I'll say it's an inducer. It is an inducer. Lactose is acting as an inducer. But what type of inducer? What is it inducing? Inducing the transcription. If operator region is free, the RNA polymerase can come over here, do the transcription, you will get all the enzymes. So this is inducing what do you call transcription. But you might have a question, how did lactose get entry? We did not increase the permeability. For increasing the permeability, you need permease, right? So always remember, even though we say it's in a switched off condition, still some amount of enzyme is produced, which is why there is permeability, which is why there is entry of things as well. Okay, I hope you got it. So it's not always lactose, it can be allolactose also, different form of lactose, that is allolactose also, that acts as an inducer. Now the lac operon consists of regulatory and structural genes arranged in the following order: promoter, then I gene, then promoter, then operator, then structural genes, that is ZYA. This is the order. Now you have been asked what is the role of lactose. So lactose help in the regulation. How? In the absence of lactose, the repressor protein is binded to the operator, it blocks the RNA polymerase, does not allow transcription to happen. In the presence of lactose, the opposite of it is going to happen. You have to write it down. And that's how you get your 5 marks, if you write all of it. Okay? Yes. Now, genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. So gel electrophoresis is being done, and then and then visualization of DNA fragments. So basically gel electrophoresis is done, but what do we add so that the DNA fragments are visualized in the gel electrophoresis chamber? What do you add? You add ethidium bromide. What do you add? You add ethidium bromide. And ethidium bromide, you cannot still see it. You need to put it under UV light. It will give a orangish, brick reddish appearance out there. Okay, the bands. Now, dash process is done using DNA polymerase, and then you get dash. Dash process is done. What is the name of the process? PCR, polymerase chain reaction. You use DNA polymerase for what? Using PCR, what do? You amplify. Amplification of the DNA. So if at all you see the first one, you get to understand what is being asked. The process, what is being used, what is being seen. So gene gun method, and what do you use for that? And transfer of recombinant DNA is basically happening. So gene gun is a method where basically you do not need vectors to transfer your DNA. Usually in the case of plants, we need agrobacterium tumefaciens, right? Suppose in gene gun method we are using it, we do not need agrobacterium tumefaciens. Since plants have a cell wall, you cannot directly inject anything. You need to have a gun, and this guns will have bullets made up of what? Tungsten or gold. Gold or tungsten filaments will be there. You will cover up it with your DNA, and then you will do the gene gun method. This is also known as biolistic. This is also known as biodistic method. And now you basically transfer the recombinant DNA. You basically transfer the recombinant DNA, but what are you using over here? Gold and tungsten filament. Gold or tungsten filament. Got it? Yes, ma'am. Now, isolation of DNA. Isolation of DNA, my dear children. How do you do it? Isolation of DNA, and then you will see precipitation of DNA. What do you add over here? You can add chilled ethanol. You can add chilled ethanol. When you add it, you will see precipitation of the DNA. DNA will be separating out. So your answer for this question is right over here. You will have it, not worry. Okay? Yes. Next. So this is for the visually challenged students only, but I have anyway covered it. Change in the frequency of genes and alleles is due to mutations, gene flow, both A and B, neither A and B. So you know the answer. It is both mutation and the gene flow. Mutation means any changes in the genetic, so obviously it is going to change the alleles. Okay, you might find new alleles being found. Gene flow is basically, suppose I am in India, someone from Africa came over here. They came along with their genes also. So allelic frequency which was there, it is being tampered. Something new is also coming over here. So obviously there is a frequency change in the case of allele. So I'll say both A and B is your answer. By the way, children, if at all you have not hit on the subscribe button, do it right now, because you are going to lose a lot of things if at all you do not. Okay, so many good things are here for you, being done for you. Make utilization of that. Yeah. Now, differentiate male heterogamety from female heterogamety. That is another question. Question number 23 for the visually challenged children. So male heterogamety means when the males produce two types of gametes. For example, you might have seen in the case of human beings, females produce XX type of gametes. Males produce two different type of gametes, either X or Y. So this is seen in the case of human beings and also in the case of Drosophila. Now there is one more condition, XXXO. This is seen in the case of insects, where again male is producing either X chromosome or having nothing in the cell, or having no sex chromosome in the cell. So two type of gametes, one with X chromosome, one with nothing, no chromosome, no sex chromosome. Okay, that is seen in the case of insects. But anyway, males are producing two type of gametes which you call as heterogametic nature. Heterogametic as in two different things. Okay, now females, do they also have this? Yes. So females, as in the case of birds, the only example that we have is birds. In them we have ZZ, but ZZ is for males. Males produce two same type of gametes, but females, my dear children, produce two different type of gametes. One is Z, other one is W. So W for women, you can remember like that. Here you see female heterogamety. So male produces two different type of gametes in male heterogamety, female produces two different type of gametes. Now male heterogamety, female produces only one type of gamete, there male produces ulta, you have written, that's all. Now sex of the offspring is decided by the male gamete, isn't it? The sex of the offspring is decided by the male gamete. What does it give? Either X or Y. Right, but over here the sex of the offspring is decided by the female gametes. Seen in the case of humans and most mammals, seen in the birds, butterflies and some reptiles. You just need to write birds, that's more than enough. Here instead of writing most mammals, you can write Drosophila and also some other insects such as grasshopper, because that is the example given in your NCERT. Okay, and gives an example, XXXO type, XXXY type, and there is ZZZW type, and mention what is ZZ stand for, what is ZW standing for. Now moving ahead with the last question, explain thalassemia as an example for Mendelian disorder. So we have two types of disorders, Mendelian and chromosomal. Mendelian means suppose there is a gene, and there's a chromosome, in that one gene is being affected, and because of that, a gene being affected, I have a problem. That is Mendelian disorder. Chromosomal means suppose I have lost a chromosome, I have gained an extra chromosome, there's some structural deformity in the chromosome. That is what you call as chromosomal disorders. So thalassemia, my dear children, it's a autosomal recessive disorder. As in, both the parents, if at all, they have at least one copy of the problematic allele, and the child gets both the copies. In the recessive condition, recessive genes only show up when both of them are same, right? So they can be seen only in the recessive condition. So if at all you see up over here, alpha thalassemia, okay, we have, hemoglobin, if at all you take as a whole. If at all you take hemoglobin as a whole, it is made up of alpha chain and beta chain. Alpha chain as well as beta chain, right? Now, always understand, alpha globin gene are located on chromosome number 16. What? On chromosome number 16. Beta globin genes are located on chromosome number 11. Over here you have two alleles for beta globin, either this or this. Suppose this is not being produced. This gene has some mutation, which is why this particular allele is not being produced, or this allele is not producing what the protein out there. Still the other one is there. Still the other one is there. It's okay. The amount of beta globin being produced will be less, but it will be still there. Suppose both of the alleles are damaged, then beta globin chain is not going to be produced. The quantity of it is going to be reduced. It is not going to be produced. Can you make hemoglobin without it? No. So that is the problem. That's when you say, yes, someone has thalassemia. It's a major patient of thalassemia. Now, in alpha thalassemia, you have two genes, and two genes have two alleles as well. Suppose this allele is affected. Still, you have three of the alleles to code for alpha globin chain. Okay, quantity is reduced, but it's still there. Suppose both of them. Then still, it is being produced because you have two more left. Suppose these two are also found. Alpha globin chain is not going to be produced at all. The quantity of it is reduced to nil. You get it? So basically, it's a quantitative disorder. The quantity is getting less of the alpha or the beta globin chain. So thalassemia is an autosomal ringed recessive blood disorder transmitted from parents to offsprings when both the parents are heterozygous carriers. Both of them are carriers. So suppose, my dear children, I assume that HB A and HB small a is there in one parent. This small a shows that my gene is defective. Similarly, I have the other parent, so I am writing over here. The other parent is also like this. Normal gene is there, but abnormal is also there. Suppose this and this comes together, won't we have HBA? And this is going to show what? The disease. This is going to show the disease out there. Okay? Got it? So if they are heterozygous carrier, the alleles are different. If they are carriers, my dear children, they can definitely pass into the children because it can be only seen in recessive condition. So thalassemia is an autosome linked recessive blood disorder, and it is transferred from both the parents to the offsprings if both of them are heterozygous carriers. Disorder occurs due to mutation or deletions of gene involved in hemoglobin synthesis, leading to reduced rate of synthesis of alpha or beta globin chains. The alpha globin chain HBA1, HBA2 are located on the chromosome number 16, and mutation or deletion of any one or more of these genes reduces alpha globin production. Similarly, the beta globin chain is located on the chromosome number 11, and mutation in one or both the alleles affect what? Beta globin synthesis. Reduced globin chain synthesis results in abnormal hemoglobin formation and anemia, which is a characteristic of thalassemia, which is what? A characteristic feature of thalassemia. Got it? Yes. That's all. So with that, we end up today's session. I hope this particular video was helpful for you. You understood each and everything in the model question paper. If at all you have any doubts that is still persisting, you can put it down in the comment section below. Wishing you all the best. Please take care of yourselves. Bye-bye.